Good morning everyone. Today we are going to discuss about biological classification. So early man classified the living organisms based on his needs, that is food, shelter, cloth, etc. So here Aristotle was the first to classify the living organisms based on simple morphological characters. So he classified the plants into three groups, that is herbs, shrubs, trees. He classified the plants into three groups, that is herbs, so into trees, herbs and shrubs. So Aristotle, so for the first time Aristotle was the earliest to attempt a more scientific basis for classification. Very important. So first scientific basis for classification given by the Aristotle, that is he classified the plants into three groups, herbs, shrubs, trees, and he classified the animals into two groups, that is those which had red blood and those that did not had red blood. And later on, in Linnaeus time, that is Carolus I Linnaeus, C.V. Linnaeus, Carolus I Linnaeus. So C.V. Linnaeus proposed two kingdom system of classification. Two kingdom system of classification given by the Carolus I Linnaeus, that is kingdom Vegetabilia, kingdom Animalia, or kingdom Plantae, or kingdom Animalia. And later on, three kingdom system of classification given by Ernest Haeckel. So three kingdom system of classification given by Ernest Haeckel. Later on, four kingdom system of classification given by Copeland. Later on, five kingdom system of classification given by the R.H. Whittaker. In this chapter, we are going to deal or discuss R.H. Whittaker five kingdom classification, that is kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia. Right? So here, so two kingdom system of classification has some drawbacks. It had some drawbacks. Now let us discuss about the drawbacks of this two kingdom system of classification. So here, this two kingdom system of classification, did not distinguish between the eukaryotes and prokaryotes. It is the first drawback. It did not distinguish between the eukaryote and prokaryote, as well as unicellular and multicellular organisms. Photosynthetic and non-photosynthetic. So photosynthetic organisms, algae, and non-photosynthetic organisms, fungi. So classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. So besides gross morphology, a need was also felt for including other characters like cell structure, nature of wall, mode of nutrition, habitat and methods of reproduction, evolutionary relationships, etc. Okay. So here in this chapter we are going to discuss about five kingdom system of classification. That is this kingdom proposed by the R.H. Whittaker. R.H. Whittaker. So five kingdom system of classification given by R.H. Whittaker in the year of 1969. So here this table is very important. So here, based on the characters like cell type, cell wall, nuclear membrane, body organization, mode of nutrition, and evolutionary and as well as phylogenetic relationships. So he classified the living organisms into 5 kingdoms, that is kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae, kingdom Animalia. So cell type, so except kingdom Monera, remaining all are eukaryote. So kingdom Monera is a prokaryotic organism, prokaryotic organism, and remaining all are eukaryote. And kingdom Protista is a unicellular eukaryote. Kingdom Protista is a unicellular eukaryote, and remaining all are multicellular eukaryotes. So in the kingdom Fungi, except yeast, all are multicellular eukaryotes, right? So next, based on the cell wall, so in kingdom Monera, mainly the cell wall is non-cellulosic in nature, so that is it made up of polysaccharides as well as amino acids. Kingdom Protista, cell wall is present in some organisms. And kingdom Fungi, so chitin is a main cell wall composition of fungi, and as well as Plantae, cellulose is a main component in Animalia. So cell wall is absent. Cell wall is absent, right? So except kingdom Animalia, we'll find cell wall in almost all kingdoms, but there are some exceptions. So exceptions గురించి మనం ఫర్దర్గా కింగ్డమ్స్ డిస్కస్ చేసినప్పుడు అందులో ఎక్కడెక్కడ సెల్ వాల్ ఉండదు డిస్కస్ చేస్తాం. ఓకే, నెక్స్ట్ న్యూక్లియర్ మెంబ్రేన్. ఇన్ కింగ్డమ్ Monera, nuclear membrane is absent. So whereas in all kingdoms except kingdom Monera, nuclear membrane is present. And body organization, so this is the NEET 2025 question. So this body organization is NEET 2025 question. So kingdom Monera, so the body organization is cellular level, and kingdom Protista cellular level, whereas Fungi multicellular loose tissue, and Plantae tissue or organ, Animalia tissue, organ or organ system. So mode of nutrition, kingdom Monera, so it shows most extensive diversity in nutrition, that is, so they are autotrophs, heterotrophs. So autotrophs again, bacteria are photoautotrophs and chemoautotrophs, photoheterotrophs, chemoheterotrophs. So they are saprophytic and parasitic, etc. And kingdom Protista, autotrophic and heterotrophic, whereas kingdom Fungi exclusively heterotrophic in nature, they may be saprophytic or parasite. And kingdom Plantae, autotrophic in nature, and Animalia heterotrophic in nature, right? So this is the table of 5 kingdom system of classification, which was given by the R.H. Whittaker. R.H. Whittaker. So R.H. Whittaker proposed 5 kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, Animalia. So the main criteria, very important. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. So cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. So here the table gives a comparative account of different characteristics of the 5 kingdoms. And the next kingdom, that is the 3-domain system. So this 3-domain system proposed by Carl Woese. So 3-domain system is divided into 6 kingdoms. So this 3-domain system proposed by Carl Woese. And here, so the 3 domains are the Archaea, Eukarya, Prokarya. Archaea, Eukarya, Prokarya. And again, this 3 domain is divided into 6 kingdoms. So you learn these 6 kingdoms about in your higher classes, right? And let us look at this 5 kingdom classification to understand the issues and considerations that influenced the classification system. Earlier classification systems included earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms under plants. So the character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. So this placed together groups which widely differed in other characters. So it brought together prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. So it also grouped together the unicellular organisms and the multicellular. For example, chlamydomonas. For example, chlamydomonas and spirogyra. These were placed together under algae. So the classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristics difference. Their walls composition. The fungi had chitin, and here in their cell walls, fungi had chitin in their walls, and while the green plants had a cellulosic cell wall. Fungi had chitin and green plants has cellulosic cell wall. So when such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. So all prokaryote organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. So in the kingdom Monera, all the unicellular prokaryotes were placed, and in the kingdom Protista, all the unicellular eukaryotes are placed, and remaining kingdoms, that is kingdom Fungi, kingdom Plantae, kingdom Animalia, all the multicellular eukaryotic organisms were placed. So kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants, and both having cell walls. So usually they are the algae, green algae. So kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants, both having cell walls, and with the paramecium and amoeba, so which were earlier placed in the animal kingdom which lack cell wall. Okay, so all these are placed in the kingdom Protista. So it has put together organisms which in earlier classifications were placed in different kingdoms. So this happened because very important. So this happened because this happened because the criteria for classification changed. The criteria for classification changed. So this kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. So depending on the improvement in our understanding of characteristics and evolutionary relationships over time, an attempt has been made to evolve a classification systems which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, that is based on evolutionary relationships. So one organism that is in ఒక సైంటిస్ట్కి ఒక ఆర్గనిజము అనిమల్ లాగా కనిపిస్తే ఇంకొక సైంటిస్ట్కి ప్లాంట్ లాగా కనిపించవచ్చు. సో దట్ ఈస్ మెయిన్లీ డ్యూ టు ద phylogenetic or is based on evolutionary relationships, and this understanding may change in future too. Okay, right. So now let us discuss about the 5 kingdom classification. Let us start with the kingdom Monera. Kingdom Monera. So very important NEET point of view as well as IP point of view. So here kingdom Monera bacteria are the sole members. Bacteria are the sole members. Bacteria are the sole members. So bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. They present everywhere. They are ubiquitous in nature. So here bacteria are ubiquitous in nature. That means they present everywhere, omnipresent. Omnipresent. They present everywhere. So, so here they are the sole members of the kingdom Monera, right? So here they are the most abundant microorganisms. So bacteria occur almost everywhere. That is why they are known as ubiquitous in nature, also called omnipresent. And the term bacteria was given by Ehrenberg. The term bacteria was given by Ehrenberg. Term bacteria was given by Ehrenberg. And here hundreds of bacteria are present in handful of soil. Not only hundreds, you'll find many number of bacteria in soil. So they also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. So that means they are omnipresent. They present everywhere, right? So bacteria are grouped under bacteria are grouped under four categories. Four categories. So this four categories was given by the E.J. Cohn. So E.J. Cohn. So bacteria are grouped under four categories based on their shape. That is spherical coccus, that is cocci, and rod shaped and spiral shaped, right? And here comma shaped, right? So the spherical coccus, cocci, which is spherical round in shape, bacilli rod shape, and we will find some spores are present on the surface of the bacteria of bacilli. And here the comma shape vibrium, that is vibrio bacteria, and the spirillum, spiral spirillum. So and these are the flagellum. These are the flagellum, right? So here, so based on the shape, bacteria are classified four groups: cocci, bacilli, spirilla, vibrio. And in the cocci, we will find again monococcus, diplococcus, streptococcus, and sarcinae. So we'll find different types of cocci that we are going to discuss in the cell, the unit of life, right? Right. So this point is very important. So this statement asked many time in the examination. That is, so here bacteria. So bacteria structure is very simple. Bacteria structure is very simple, and they are complex in behavior. They are complex in behavior. So structure is very simple and complex in behavior compared to many other organisms. Many other organisms. And here the bacteria show most extensive metabolic diversity. Very important. Most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food. They synthesize their own food, that is autotrophic. And they may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast, very important, the vast majority of bacteria are heterotrophs. the vast majority of bacteria heterotrophs. So they are simple in structure, complex in behavior, and they show most extensive metabolic diversity. Most extensive metabolic diversity. So these points are very important for a NEET point of view. Right. So then come to the archaebacteria. So in the archaebacteria, so these bacteria are specialized since they live in some of the most harsh habitats such as salty areas. Very important. So from this area, so NEET exam లో చాలాసార్లు అడిగారు ఈ క్వశ్చన్ ఆర్కిబ్యాక్టీరియా నుండి. సో హాలోఫైల్స్ మీన్స్ సో బ్యాక్టీరియా విచ్ కెన్ లివ్ ఇన్ extreme salty areas. They can tolerate extreme salinity and hot springs. So the bacteria which grow in hot springs called as thermoacidophiles, and the bacteria which grow in marshy areas called methanogens. And archaebacteria differ from other bacteria in having different cell wall structure, and this feature is responsible for their survival in extreme conditions. So this statement was given many times in the examination. So different cell wall structure, archaebacteria having different cell wall structure, due to that it can survive in extreme conditions. It can survive in extreme conditions. And methanogens are also present in the gut of several ruminant animals such as cows and buffaloes. So and they are responsible for the production of methane, that is biogas, from the dung of these animals, that is excreta of cows and buffalo. Okay, so this is about the archaebacteria. So we have to remember the halophiles, thermoacidophiles, methanogens. Halophiles means archaebacteria which can survive in extreme conditions, extreme salt conditions, and thermoacidophiles means bacteria which can live in high temperatures, hot springs, and methanogens means marshy areas. And these bacteria having different cell wall structure, that is why it can survive in extreme conditions, and it possess branched chain lipids in their cell membrane. Archaebacteria possess so branched lipids in their cell membrane, in their cell membrane, and having the different cell wall structure. Due to that, it can survive in extreme conditions. And here this methanogens also present in the, that is, gut of several ruminant animals. Those ruminant animals are cow and buffalo, right? So in their excreta, so here the methanogens are present, and they are responsible for the production of biogas, also called methane gas, right? So it is also called as gobar gas. It is also called as gobar gas. And now let us discuss about the uh, eubacteria. Eubacteria. So this is a rough diagram of eubacteria. The first layer is a glycocalyx, and cell wall, glycocalyx, cell wall, and cell membrane. Cell membrane. Right. So here this cell membrane have some projections inside towards the semi-fluid matrix, that is cytoplasm. called as mesosomes. Mesosomes. And it is the outermost layer is known as glycocalyx, and this blue color line is known as a cell wall, and this black color line is known as the cell membrane or plasma membrane. And the structure of prokaryotic we are going to discuss in Cell the Unit of Life in detail. Okay, so here the mesosomes are found in the form of lamellae or tubule form or vesicular forms. And here um, the main genetic material of this prokaryotic structure is a nucleoid. It is naked. Generally, the main genetic material of prokaryote is naked. That is called as nucleoid. It is also called as genophore. Nucleoid is also called as genophore. And along with this naked genetic material, extra genetic material is present. That is called as a plasmid. That is called as a plasmid, or we can call it as episome. Episome or plasmid. And a membranous bound organelles, ribosomes are found. Ribosomes are found, and they have specific functions. Mesosomes have specific function, ribosomes have specific functions, and plasmids have specific function. So all those things we are going to discuss in the Cell the Unit of Life. So the main function of ribosomes is they involved in the protein synthesis, and mesosomes function is respiration, and plasmid function. So they are used as cloning vehicle. They are used as vectors in RDNA technology, right? So we have different functions. So all these we are going to discuss in the Cell the Unit of Life, right? So here, if you talk about the majority of the bacteria are heterotrophs. So majority of the bacteria are vast majority of the bacteria are heterotrophs, and here the majority are important decomposers in nature. Majority are decomposers, and here they are helpful in making curd from milk and production of antibiotics and nitrogen fixing in legume plants. So these are some of the important uses of heterotrophic bacteria. So uses of heterotrophic bacteria, that is, so making curd, uses of heterotrophic bacteria. So here troph means nutrition. So here, so first use is the production of curd from milk and production of antibiotics. So these antibiotics are mainly produced from a group of actinomycetes, that is specifically streptomycetes. So example is streptomycin, and nitrogen. Another use is the nitrogen fixing in soil. So here some bacteria can fix nitrogen freely in the soil. Some can fix in symbiotic association with the legume plants, right? So these are some of the uses of this heterotrophic bacteria. And the most important thing of this mode of nutrition, chemosynthetic autotrophic bacteria, very important. So chemosynthetic autotrophic bacteria, so they play an important role in mineral recycling. That is chemo autotrophs. Chemo autotrophs, they play an important role in mineral recycling. Mineral recycling, such as nitrogen, phosphorus, iron, and and sulfur. Okay. So this is the important point, right? And next, this is about the diagram given here is a cyanobacteria. Cyanobacteria. So here, if you observe the cyanobacteria, so this bacteria is surrounded by a mucilaginous sheath, or it is also called as gelatinous sheath. And this cyanobacteria is also called as blue-green algae. So here, right, the cyanobacteria, also referred to as blue-green algae, have chlorophyll A, very important, similar to green plants. Similar to green plants. That is, they are photosynthetic in nature. So the cyanobacteria are unicellular, colonial, or filamentous, freshwater, marine, or terrestrial algae. They may be unicellular, colonial, filamentous form. And here they often forms blooms in polluted water bodies. That is, algal blooms. Algal blooms. So here, so if you take a polluted water body, for example, this is the polluted water body. Polluted water body. So initially there is only one cyanobacteria or one blue-green algae. It undergoes rapid multiplication. It undergoes rapid multiplication. Cyanobacteria undergoes rapid multiplication, utilizing the nutrients from that pond, and so it divides by binary fission and produces so many progenies. 1 become 2, 2 become 4, 4 become 8, 8 become 16, 32, like that. It undergoes rapid multiplication, and here it expands entire pond. It expands entire pond. So make it as a dense mat. So it appear as a thick, dense mat. Okay, and here that is nothing but cyanobacteria. Cyanobacteria. So due to this thick, dense mat, or it is also called as colored scum. Colored scum. So due to this, sunlight cannot penetrate into the pond, oxygen cannot penetrate into the pond. Okay, so that the organisms live in this pond, which depend on sunlight or oxygen, so they will die. Okay, so this is called as a algal bloom. This is called as a algal bloom. So usually they form blooms in polluted water bodies, not in fresh water bodies. They form algal blooms in polluted water bodies. And another important thing is cyanobacteria possess specialized cells. Specialized cells. So these specialized cells known as heterocysts, so which involved in the nitrogen fixation in soil. Nitrogen fixation in soil. So that soil become fertile. Further, it leads to the improvement in crop yield. So examples for this cyanobacteria, that is Nostoc, Anabina, oscillatoria. So this is about the cyanobacteria, and right. So here again some points are given regarding the eubacteria or bacteria. Eubacteria means true bacteria. It is also called as bacteria. So here some pathogens causing damage to human beings, crops, farm animals and pets, cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. So cholera is caused by the Vibrio cholera and typhoid is caused by Salmonella typhi. So cholera is caused by Vibrio cholera and typhoid is caused by Salmonella typhi. Tetanus is caused by Clostridium tetani. Clostridium tetani. So citrus canker is caused by Xanthomonas oxynopodis pathover citri. Okay. So bacteria reproduce mainly by binary fission. Binary fission, that is asexual mode of reproduction. Asexual mode of reproduction. Sometimes under unfavorable conditions, very important. Sometimes under unfavorable conditions they produce spores. Spores. So they also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to other bacterium, that is nothing but genetic recombination. That is, so true sexual reproduction is absent. True sexual reproduction is absent. So true sexual reproduction is absent in uh, bacteria, but the sexual reproduction occur by means of genetic recombination. Genetic recombination. And the mycoplasmas are organisms that are completely lack the cell wall. Mycoplasmas are also called as PPLOs.